Okay, this is the third lecture on the chapter on lipids. We've covered fatty acid, prostaglandins, waxes, and triglycerides. Next we are going to cover, there you go. We're going to cover glycerophospholipids and sphingolipids, which come in, I guess, if you wish, two flavors, sphingolipids and then glycosphingolipids. The main difference here is they have carbohydrate attached to them, so we'll call that number 3. All right, so glycerophospholipids, as the name suggests, they have glycerol, right, fatty acids, and then phospho for this phosphate. So it's similar to a triglyceride, where we had fatty acids esterified to these. Right, so one fatty acid 1, fatty acid 1, fatty acid 2. These are ester linkages, so remember this is a fatty acid, so these are esters. So ester linkages, which means of course these are saponifiable. Now down here, instead of having a fatty acid ester, we have a phosphate ester. So let me just go ahead and draw the structure of this phosphate. I'll put that up here. This is actually, right, so phosphoric acid, right, is an acid. And what does this phosphate look like? Right, so this is the phosphate polyatomic ion. Hopefully you're familiar with that. Phosphate, PO4-3. And if you remember, the fatty acid, carboxylic acid, I'll put that here. Right, so it has a long hydrophobic tail. Right, so you have a carbon double bonded to an oxygen and then O-. Here we have a phosphate double bonded to an oxygen and then it's O-, O-, O-. Right, there's 4 oxygens, a -3 charge. So what we've done here now is we've esterified glycerol. Remember, glycerol is an alcohol. Right, an ester is a mixture of an alcohol and an acid, right? So here we have a fatty acid, here we have phosphoric acid. So we're going to stick glycerol, stick another color here. So we're going to stick a glycerol onto one of these, right? Just as we would here with the fatty acid, we'll stick a glycerol and an ester linkage to that fatty acid. But then we also have this thing, it's an amino alcohol. So I will stick an amino alcohol, amino alcohol, it's clearly written down here, amino alcohol to this one. This carbon, or this rather oxygen here, is not esterified. Not esterified. That's spelled right, but anyway, so we're going to have phosphoric acid is a triprotic acid. You could, I guess, potentially have 3 esters. Fatty acids are a monoproduct, so just one alcohol group esterified. Here we're going to have 2. We're going to have a glycerol and this amino alcohol. So what is an amino alcohol? And actually, before I get onto that, what I was going to talk about here is this is not esterified, so notice it has a -1 charge. So I'm going to go ahead and put an oxygen here with this -1 charge. This is going to be important later. Right. So glycerophospholipids are polar, right? And the reason for that, remember we have, let me just draw ahead, we have the glycerol, we have a fatty acid here, a fatty acid, fatty acid here, and that's really not really any polarity here because again it's an ester. But this guy here has the phosphate, remember, with that negative charge, and then an amino alcohol. And that amino alcohol is polar. So we've got some polarity here, right? We have polarity from the negative charge and also from this amino alcohol. If you look at these amino alcohols, there's 3, choline, ethanolamine, and serine. Let's just look at, let's look at ethanolamine for starters. Ethanol is a 2-carbon alcohol. So right here is ethanol, and all we've done is we've put an amine on it. Now, under physiological conditions at physiological pH about 7.3 or 5 or so, this amine is going to be protonated. So remember, ammonia is a neutral compound, NH3. Normally, nitrogen forms 3 bonds and it's neutral, but it can gain a hydrogen and form the ammonium ion, right? So it has a plus charge. Well, here in ammonia, right, this nitrogen has 4 hydrogens attached to it, so 4 bonds. This nitrogen has 4 bonds, only 3 hydrogens, but it's also bonded to this carbon, so 4 bonds. It has a plus charge. Right? This is the alcohol. This is what gets esterified to the phosphate. Well, I'll go ahead and put -3, but this is the alcohol portion that gets esterified to this acid. Here we have an alcohol, in choline gets esterified to the phosphate. Here in serine. Now, serine is actually an amino acid. This is rather interesting. This is an alpha amino acid. It's the next chapter, actually. Alpha amino acid. So it has an acid functional group, right? It has the amine. So a positive, a negative, and then this alcohol, of course, then gets esterified to, just go like this. This is esterified to the phosphate. Right? Phosphate's an acid. You make an acid by combining an alcohol with an acid. You get an ester. So this part isn't polar, but then we have the positive and negative charges here on serine. This is a quaternary amine. Right, again, it has 4 bonds, and so it has a plus charge. Physiological pH, again, ethanolamine is going to be charged as well. So these guys, not only are they polar, they actually are charged. They're ionic. Okay, so quite polar. So here they've shown the 2 fatty acids. Right, so fatty acid 1, fatty acid 2. This one has a double bond, so there's a kink in it, right? It's a cis fatty acid. Here are the esters, so they're esterified to the glycerol backbone. Right. The amino alcohols are these green guys, right? And there's 3 types, remember? Oops, I wanted to use green. There we go. We had ethanol, which is plain old ethanol, but then we stick an amine on that other carbon, so ethanolamine. Serine was that amino acid, so it had the positive and the negative charge. And then lastly, choline. Choline was the one with the quaternary amine. So that's, and in fact, that's what this is here. This is choline. In fact, right here, choline. 1, 2, 3, 4 carbon nitrogen bonds. This is a quaternary amine. So unlike triglycerides, which were essentially, they were oils or nonpolar, this guy is what we call amphipathic. And basically what that means, it has 2 different polarities. So part of the molecule is polar. That's what we call this polar head group, made up of the negative charge in the phosphate. Remember, this is that one oxygen that is not esterified, so it's got a negative charge. And then the amino alcohol, which has at the very least a nitrogen with a positive charge. Of course, serine has both the positive charge on the nitrogen and the negative charge for the acid. It's an amino acid. So this is the polar head group. So part of it is polar, that's the head group, made up of the phosphate and the amino alcohol, and then we have part is nonpolar, and that is these esters here of glycerol and the fatty acids. So I'll just say glycerol ester, I shouldn't say of, and the fatty acids. All right, so just a little bit of nomenclature here. Lecithin, right, is, well, another name for lecithin is phosphatidylcholine. So this is phosphatidylcholine. So when you have a phosphate, glycerol phosphate, that's a phosphatide. We had like glycerides, this is a phosphatide, and when it contains choline, it's lecithin. So lecithin here has choline. Acetylene contains ethanolamine. Now, one thing that you might be somewhat familiar with, choline is a neurotransmitter. So it has something to do with nervous tissue, and cephalin, you think of maybe cephalic or cerebral. These guys are also involved in nervous tissue, and they're abundant in brain and nervous tissue. Quick learning check here if you want to pause and see if you can answer these. Identify each as one of these. So a glycerol trioleate. So this, or glycerol. So this means we have glycerol, right? And tri means 3. We have 3 oleic acids. That's what oleate is. It's a conjugate base of oleic acid. So this is a glycerol and 3 fatty acids. Well, that's a triacylglycerol. So that's B. Cephalin, that's a glycerophospholipid. Right, that's D. That's a glycerophospholipid, and I'll just put here that's the one that contained ethanolamine. Choline, right? Choline is found in glycerophospholipids, but all by itself it is not. It's just plain. It's just a simple amino alcohol. So that's C. And palmitic acid is simply a fatty acid. So A. Some more practice here. So identify the components and type of glycerophospholipid. All right, so let's use different colors here. So for blue, this is a glycerol backbone. All right, so that's glycerol. We have here 2 fatty acids. It's 16, 17, 18 carbons long, and this is saturated, so this is stearic acid. So we have fatty acid 1, fatty acid 2. Then we have a phosphate. I'm going to use yellow for phosphate, and the reason for that is typically phosphorus is color-coded yellow. So we have a phosphate. Hopefully you can see that. And then lastly, we have this. What is this? This is an amino alcohol, and in fact this is serine. Serine, which is not only is it an amino acid, but so we got the amine, we have the amine, we have the acid, and then the alcohol, you don't see it in OH here because it's in an ester that's been esterified to the phosphate, but this is the amino alcohol. Okay. So this is a cephalin. Cephalins are glycerophospholipids. It says type of glycerophospholipid. Glycerophospholipids that contain serine or ethanolamine are cephalins. Lecithin is the one that contains choline. So we talked about these glycerophospholipids. Oh, one thing I forgot to mention, right? This has got the, okay, so this phosphate has a negative charge. The amino alcohol could be positive or negative. So we have this polar head group, and then 2 fatty acids coming off it. Remember, the polar head group contains the phosphate and the amino alcohol. The fatty acids and the glycerol esters here are the tails, and it's amphipathic. So this is polar. This part's non-polar. And these are found primarily are the components of cell membranes. So right then, these are components of cell membranes. All right. Next, we'll talk about the sphingolipids. Kind of a funny name, sphingolipid. And by the way, I mentioned here, these are not saponifiable because there's no esters. Well, they kind of, well, you'll see this one kind of does have an ester, but we'll get to that. Okay, so sphingolipids. They're similar to phospholipids. Why is that? Well, they have a polar part. Remember, this is a polar head group of phosphate and amino alcohol. So this is polar. Sphingosine here, this is an ester. And this is also an ester, just as it was in glycerophospholipids. This, however, is not. This is not an ester. And saponification, you remember, if you recall, is soap making. So what makes a soap is the sodium salt of this fatty acid, which means it has to be linked in an ester. Well, this is not in an ester, so it's not saponifiable. But it is amphipathic, right? It has a polar and a nonpolar region. We call this amphipathic. Amphipathic. Okay, let's look at the structures of these next. So sphingosine is this long chain amino alcohol, right? So we've got this, kind of looks like a fatty acid, long chain here. And then it has an alcohol, an amine, and an alcohol. So remember glycerol. So this kind of forms the backbone like glycerol does. So this is sphingosine is the backbone of sphingolipids. Right? Just as glycerol is the backbone to which we'll esterify or link things to. It's the backbone of triacylglycerols, or triglycerides, same thing. But then also we talked about glycerophospholipids. Glycerophospholipids. Okay, so it's got this backbone. We have an alcohol group, an amino group, and an alcohol group. Now remember, glycerol, it was just 3 carbons, and it had 3 alcohol groups. And you could stick 3 fatty acids on here, or maybe 2 fatty acids. In the glycerophospholipid, you have 2 fatty acids, a phosphate, and then amino alcohol. So this is kind of the business end. This here, this is that long chain non-polar part, so it's non-polar. But this is the business end, right? So let's see how we're going to decorate this backbone in a sphingolipid. So here's that long hydrophobic tail. Notice this alcohol group here is not used. Right? So, like with glycerol, we had a fatty acid attached to each, but here we're not going to use this alcohol, and instead we use this amine. Right? Well, this is not an ester. This is an amine plus an acid. If you remember that chapter on carboxylic acid, or sorry, not carboxylic, on amines and amides, this is an amide. If you take a carboxylic acid, like a fatty acid, and combine it with an amine, you get an amide. And therefore, this is not saponifiable. Not saponifiable. Okay. So the fatty acid is linked in an amide linkage, not an ester. The last alcohol on the bottom, remember, was alcohol, amine, and then another alcohol. So we have the phosphate and here choline. So again, a negative charge and a positive charge. This is the polar region. So this is like the polar head group. When you have a sphingolipid that contains choline, it's referred to as a sphingomyelin. and this forms the myelin sheath that's found around nerve cells. If you want to think of the myelin sheath kind of like insulation around electrical wiring, so that's what these do. So they're found in nerve cells, and sphingomyelin forms the myelin sheath around nerve cells or neurons or just nerves. Okay, so that's sphingomyelin. It's a sphingolipid that contains choline. Glycosphingolipid. Glycosphingolipids, again, as the name suggests here, mention this right at the top, glyco, like glycogen, this is a carbohydrate. So we're going to have a sphingolipid, and then, actually let me go ahead and put the alcohol here, right? This here is the amide. This is also an alcohol. Right? So remember we had an alcohol, an amine, a second alcohol. So here we have a, well, this is a monosaccharide, so a carbohydrate bonded to sphingosine. What kind of bond is this where we have a carbohydrate bonded to an alcohol? Remember that? That's that glycosidic bond. Right? This is, in other words, this is an acetal. So those goofy acetals have reared their ugly head again. So in more detail here, right, here's the long hydrophobic tail. Hydrophobic, or if you wish, nonpolar. Remember, this alcohol isn't used. Here we have the amide and the fatty acid. Again, long chain fatty acid, also nonpolar. But then we have this acetal, right? Here it's carbon, oxygen, carbon, carbon, oxygen, carbon. So this is our acetal carbon, carbon number 1, and this is actually galactose, so it's a galactocerebroside. These are also found where? Well, cerebrosides, again, like cerebellum or cerebral, these are also found in nerve tissue. Found in brain or nerve tissue. So it turns out your brain is very fatty. So if someone calls you a fathead, it's really not an insult. Gangliosides. Gangliosides. Heard of neural ganglia? Right. So again, as the name suggests, these are found in nervous tissue, important in neurons. Neural ganglia. A ganglia is like a nerve bundle, so a gang of nerves, right? So I like to think of gangliosides, and remember we had carbohydrates that ended in -ose, so this is kind of like that. This is gangliosides. These again contain here 2 or more monosaccharides. So it's the gangliosides. I think of carbohydrates. So you can think of carbs or sugars, right? Because they're monosaccharides or sugars. And here we have a gang sugars. So we have a number of sugars, not just one, right? Not just like cerebrosides on the last page. We have now gangliosides that have more than one. And these are act as receptors on cell surfaces for hormones and viruses. So I don't know, maybe you want to do some research, find out what the binding site is for the COVID-19. I'll just here. Actually, COVID is the name of the disease caused by the coronavirus. I can't remember the name of the formal name for the virus, but put a question mark here if you want to look that up. The other thing about these is there are a number of genetic diseases where these guys accumulate. So we'll talk about that just briefly. So here's a specific ganglioside. Again, let's just focus on trying to figure out, what's going on here. Here's that sphingosine, that long tail, the alcohol that's not used. All right, so here's the sphingosine backbone. We have a fatty acid in an amide bond. We have the carbohydrate in a glycosidic bond, or if you wish, an acetal linkage. And then we have a number of sugars here. glucose, galactose, and acetylglucosamine. So what they've done here is instead of having an alcohol, it's an amine. And then acetic acid is linked to this amine as an amide, and that's this N-acetyl. The N means this nitrogen. Right? The acetyl group, this is an acetyl group. We'll talk a lot about that when we get into, sorry, that's the acetyl group. It's like an acetic acid or an acetate molecule, and it's linked to the nitrogen, so N-acetyl. Anyway, this accumulates in genetic disease called Tay-Sachs. I think on the last slide here, there's another number of other diseases. You don't have to know these diseases. I just find it interesting. A number of different diseases where these accumulate and basically cause neuronal problems. And so because these are genetic diseases, oftentimes people seek out a genetic counselor that will educate them if they'll do a genetic test and find out if they're a carrier of this disease, and if they're getting married, if their spouse is also a carrier, if they really want to try having children or not, because these diseases can really be quite traumatic. All right, go back here and review a little bit. So we talked about these glycerophospholipids. They're saponifiable. They have these esters. Right? They're found in cell membranes. We have the sphingolipids and glycosphingolipids. They have the sphingosine backbone. These are found in nervous tissue. They're not saponifiable. Not N-O-T, not saponifiable. The main difference between sphingosines and glycosphingosines are the sugars, right? Glycosphingolipids have sugars, but then we also had, maybe I'll put here, we had gangliosides, right? So this was had I'll say 2 or more monosaccharides. With the sphingosines, this amino alcohol, right? If it contained choline, right? We call that a sphingomyelin. Myelin. Okay. If it was ethanolamine or serine, it was cephalin. So ethanolamine or serine. Okay. The last thing then we'll talk about are steroids, and again I also mentioned something about, I mentioned here cell membranes. We'll talk a little bit about cell membranes in the next video.